Good morning, everyone. My name is Joanna Jurek, and today I'm delighted to present my PhD project, which is an investigation on how mercury exposure can impact cytokines IL-33 and IL-17, which have been recently associated with development of autoimmune disease. First of all, what is an autoimmunity? So our immune system is trained to does not react against self-tissue. Therefore, we have no response and no associated damage with it. What happens in autoimmunity is that the immune cells mistakenly recognize own tissue as a foreign material. Therefore, they produce autoimmune response, which is associated with increasing inflammation and also tissue damage. An autoimmune disease I'm going to focus on in my PhD is systemic lupus erythematosus, in short called lupus, which prevalence in Northern Ireland is about 254 cases per 100,000 cases. What happened exactly in SLE is that immune system of these individuals recognize own material, which is in this case genetic material DNA, and produce immune response associated with high production of antibody. Therefore, these antibodies can trigger inflammation in many systems within the body. The most common is a skin rash, called also as a malar rash, but more severe is a kidney damage, which is the main cause of mortality in lupus patients. So, what does go wrong with the immune response in SLE? So, whenever our cells die, they release intracellular material. This intracellular material, called also as rich in self-antigens, is taken up and subsequently presented by antigen-presenting cells to the Th0 cells, which are immune cells. And then these cells need to decide, depends on the self-antigen, what sort of immune response they induce. In healthy individuals, self-antigens are known as the own material. So Th0 cell became Treg cell, which main function is to switch off the immune response. Therefore, there is no immune response. What happened in autoimmunity and also SLE is that the self-antigen is recognized as a foreign material, so Th0 cell triggers the immune system. Especially in lupus, historically, the Th1, which is a cell-based response, and Th2, which is an antibody-based response, have been implicated. However, there is an increasing and accumulating evidence that actually Th17 cells can be involved in the global inflammation as well. And particularly on the cellular response, I'm going to focus in my PhD. Lupus is such diverse pathology, so there is no surprise that many factors are implicated. For example, environmental exposure to toxicants like mercury has been recently suggested as a potential effect on exacerbation of the disease. But diet and exposure to long-chain polyunsaturated fatty acids has been suggested to ameliorate the development of lupus. So how we can be exposed to the mercury? So we can be exposed through dental fillings, we can be exposed by old intramuscular injections and vaccinations, but most predominantly we are exposed to mercury, especially methyl mercury, which is an organic form of mercury, through high fish consumption. There is increasing evidence from in vitro animal and human studies that actually mercury can contribute into autoimmunity. For example, cell studies shown that that the mercury is cytotoxic to immune cells, and also exposure to the mercury can result in a high pro-inflammatory cytokine production. Most of the evidence came from the animal studies, where researchers exposed genetically susceptible animals to the mercury, and in a result, these animals were able to produce high levels of autoantibodies as well as pro-inflammatory cytokines. Human studies results are inconsistent. For example, exposure to mercury through diet in healthy individuals result in higher pro-inflammatory cytokines production as well as increasing production of autoantibodies. But exposure to inorganic form of mercury of the autoimmune patients, SLE patients in particular, has shown no effect of disease activity or damage. Interestingly, another study has shown that the main way of exposure these patients to the mercury was through the diet and fish consumption. It's important to emphasize that actually fish meat is also source of the mercury exposure, but in the same time is rich in a polyunsaturated fatty acids, which potential anti-inflammatory effect might ameliorate pro-inflammatory effect of the disease. So in my PhD, I'm going to focus on the environmental exposure through diet of a high fish consumption and see if this exposure to methyl mercury might contribute to cellular injury, cause cell damage, which is necrosis, therefore contribute to production of pro-inflammatory cytokines, but also in the same time, if during this cell death, release self-antigens might also contribute to the pro-inflammatory cytokines production, but also activation of Th17 cells, which have been implicated in autoimmunity. In the same time, by high consumption of fish, I'm going to investigate that if long-chain polyunsaturated fatty acids might actually ameliorate this pro-inflammatory effect of the mercury exposure. I'm going to address this aim in 4 different studies. First study is an observational study, and I'm going to look at the healthy population who are high customers of fish from Seychelles. And what I'm going to do is to measure their blood serum levels of IL-33 and Th17 cytokines and see if there is any association between their dietary exposure to methylmercury and levels of these cytokines. In study 2, I'm going to look at the cohort, especially lupus patients, and in these patients, I'm going to assess IL-33 and Th17 cytokines and see if in this case of autoimmune prone individuals, is there any association between mercury exposure and levels of the cytokines. Having an appreciation that this observational study has some limitation, I'm going to take the immune cells from lupus patients and healthy controls and expose them in laboratory conditions to methylmercury alone, but also methylmercury with long-chain polyunsaturated fatty acids, and see how does it impact the release of cytokines, IL-33 and Th17 cytokines, and the cytotoxicity of cells. Last study, study 4, involved a spontaneous model of the lupus, and I'm going to expose this animal to the mercury through diet, and also see I'm going to expose these animals to mercury through diet and polyunsaturated fatty acids and see if this dietary exposure to mercury might increase or affect IL-33 production, and also I'm going to see if the PUFA present in the diet can ameliorate level of the cytokine. Lastly, I'm going to see that if I block IL-33 in these animals, how does it impact of this IL-33 activity. I'm going to assess animals' blood and also take some histology samples in order to make an analysis. I hope that knowledge gained from my PhD can can be utilized by my founder, Department for Education, but also professional bodies who investigate the autoimmune disease and lupus in particular. And I hope that that can contribute to better understanding and importance of the fish consumption by lupus patients, autoimmune patients, and also will increase knowledge of the high risk customers. Thank you very much for your attention.